Hi, everybody. My name is Gladys Kaitani, and I work at Adaptive. I lead the antibody discovery group back in Seattle. So today, I'm going to kind of give you a little overview of what we've been doing at Adaptive with antibody discovery. The title of my talk is Unlocking the Value of Immune Receptors as Therapeutics. Do I use this? Okay, perfect. So before I get into antibody discovery, I wanted to just give you a little bit of overview about what Adaptive does, specifically in the immune medicine platform. As a lot of you know, Adaptive is divided into sort of two different businesses. We have our diagnostic business, which I'm not going to touch about today, and then we have the immune medicine business, which is basically the kind of the life cycle of it is that what we do is we're really good at sequencing immune receptors, both TCR and BCR. And we start with sequencing TCR in patients and that are either, have a specific disease, and then comparing that with naive patients, basically. And then we use that to figure out what are the enhanced TCR sequences that these patients have, and we make a map. And once we have that map and identify specific TCRs, we go back and do pairing of those TCR. And this is the most important part I'm going to talk about today, is pairing, and what technology do we use to pair both TCR and BCR. And once we pair the TCR, we can go back and figure out what is, we can use that for target discovery. And basically saying, what do these TCR recognize? What are the antigens? And then once we figure out the antigen, that comes to my team, and then how do we find a drug, basically an antibody against that antigen? So that's sort of our new pipeline. But before that, we wanted to validate our BCR pairing. As a lot of you know, with BCR, a lot of the time we're using single-cell sequencing. What Adaptive has done, and this was published about 5 years ago on TCR discovery, is using what we call pair-seq to pair T cells at really high throughput without using single-cell. So today I'm going to kind of show you a proof of concept data using infectious disease, and this started during COVID about 3 years ago. So how does this go? So basically, we start with, when it comes to COVID, we started with patient samples. We can do this with animal models also. Right now, we're using humanized mouse models. And basically, we isolate B cells either by using antigen tetramers, so basically doing antigen-specific sorting, or we can just isolate plasma cells. So it doesn't have to be, it can be antigen agnostic, basically. And then we enrich those B cells. We do a quick expansion in culture, because one of the things that you will see with our pair-seq is that you need a few representative of each clone to be able to pair. And then we go into our pair-seq, which I'll go into it a little bit more. And this is really high throughput. We can do millions of cells at the same time. And then we have a well-developed in-house, in silico manufacturing risk prediction with our bioinformatics team. And they basically give me some antibodies. We express those antibodies, and then we go into preclinical evaluation. So I'm going to go through a couple of examples today. So how does pair-seq work, basically? So it is quite simple. What we do is, after I expand those cells in culture, we distribute it into a 96-well plate. And it can be multiple 96-well plates. It doesn't have to be 96. That's just what's worked for us. And we do a very quick expansion so that we don't change the cells in culture. And then we go in and we sequence both the light and the heavy chain. And each one of these wells is barcoded. And then basically how we figure out what's a pair is based on statistics. What are the chances that two sequences can occur in the same well? And we want them to co-occur in the same well about 5 times for us to say that's statistically significant. And that's why I said we need to expand the cells a little bit, because we need about 5 cells basically per clone to be able to pair them. And then bioinformatics will basically spit out a bunch of sequences. They will go through a pipeline where they're selecting the best antibodies, the one without manufacturing risk. We look at things like somatic hypermutation, and then pick the antibodies that we think will be the best binders and will be a lot more functional. And then we make those antibodies, we screen them, we find basically a subset of antibodies that were like, these are functional. The real beauty of our technology is that we can actually go back to the data set, because when we're doing this, we're just picking one antibody, one representative antibody per lineage. We can go back to that data and say, okay, we found this one antibody that's really good. Let's go back to our data set and see if we can extract more antibodies without going back to sorting and sequencing again. So that's what I've really enjoyed about this, is that we've been able to pick an antibody, go back to our data set, and find an antibody that's even 10, 20 times better than the original antibody. So basically, what we've done in the last 3, or at least 2 years before this year, is that we looked at 3 different diseases. We started with COVID because that was an opportunistic moment for us to get our PSC going. And then we went to flu and RSV. Today, I'm just going to talk about the COVID program and the flu program. Okay. So with COVID, everybody kind of jumped into COVID when in 2020 when it first appeared, and adaptive. It really by May we already was setting up this program, and at the time we didn't have a PSC assay that was working for B cells. We had one for T cells, so we were learning how to do PSC for B cells as we were doing the project itself. So we took about 300 individuals. And basically sorted antigen-specific B cells from those people, and this was mostly spike and RBD. So we used the trimer and then made a tetramer out of RBD. We were able to pair a lot of antibodies and select about 100,000 of those antibodies were actually in-frame antibodies. And then we decided to make about 2,500 of those. And this was in collaboration with GenScript, who really, at speed, really high speed, were able to make these antibodies and turn around, give us antibodies within a few weeks. And of those 2,500, about 1,100 of them actually bound to SARS-CoV-2. And we found antibodies that bound to all different parts of the domain. So there were antibodies that bound to RBD, antibodies that bound to S1, S2, and there were these really interesting antibodies that only bound to trimer, and I think if you read about COVID antibodies, these were quite common, and we think that it's probably binding to different parts of each trimer. So if you take one subunit of RBD or S1 or S2, they don't bind, but if you have the trimer intact, they will bind. So those were really interesting. So basically, this proved to us that we are able to pair these antibodies using this really high throughput system, and we were able to find a very diverse set of antibodies. So we took these antibodies that bound, and we did basically a neutralization assay and selected the antibodies that bound at high affinity, that neutralized with really low IC50s. And this is jumping a lot of data that we did to characterize these antibodies. But basically what we did is we took the antibodies that neutralized, and then at the time there was this paper, Burns et al., that basically grouped antibodies into 4 different classes. So we were able bioinformatically to figure out which classes to the set of antibodies we've selected belong. And then we just selected a subset of antibodies from each class, and those became our lead candidates. And for those who are familiar with COVID antibodies, those class 2 antibodies, after the first variant, they were all gone. So that was another kind of beauty, is that figuring out that, class 1 and class 3 actually are still inhibiting these new variants, and going back to our data set again and say, can we find antibodies that are more similar to this class 1 and 3? So we were able to replace those class 2 really quickly. And then here at the bottom, we were able to put those RBD antibodies into animal models, and this is a mouse model. This was in collaboration with the COVID project that took hundreds of antibodies and tried to compare them and see which ones have function. And with our RBD antibodies, really majority of them protected mice up to 100%. And another subset of antibodies that we identified was our antibodies that were really unique, and they bound to S2. And these antibodies, I don't have a slide for this, but they came from one specific patient. And this patient was able to clear infection within 3 days upon onset of symptoms. So we had looked at hundreds of other patients, and with this one patient, it was really interesting because 90% of the antibodies bound to S2. So we knew this was unique because that was not the case with the other patients. So we selected some of these antibodies, and what was interesting is that it bound to other coronaviruses, not just SARS-CoV-2. And for some of those, when we put into animals, we did see some protection. One interesting thing with these S2 antibodies, which I think has been published in the field, is that they actually were a lot less potent. So they recognize a lot of coronaviruses, but they're not as potent as the RBD antibodies. So looking a little bit into these S2 antibodies, what was the mechanism of protection? One thing so we just performed a regular neutralization assay, and we did see that they do neutralize. It's about 100 times less than what the RBD antibodies can do. We also collaborated with Khalid and saw that they have some effective function. But what was really interesting is we wanted to understand a little bit more, the reason why they protect animals is not really because of neutralization, because that is a really low or at least high IC50. So we worked with GenScript to develop this fusion assay, whereby we had a reporter system, one cell that expresses ACE2 and another cell that expresses spike, and when they fuse, there's expression of luciferase. And it was pretty exciting because what we found was that these S2 antibodies actually block fusion of the two cells. I don't know if you can see that, but the two antibodies sort of at the bottom, the red and the orange, those are COVID RBD antibodies, and they don't block fusion as good as the S2 antibodies. So we think that these S2 antibodies have a really interesting mechanism. But the problem with S2 antibodies was later on because we developed these based on the wild type spike, so to be able to study all the other variants, we have to basically develop a new fusion assay, and we haven't done that yet. But these has been really sort of interesting antibodies to look at. So again, as I said, we collaborated with the COVID project, and they took basically our antibodies and added it into their 300 and something antibodies that they were looking at. And two of our antibodies actually belong to this group of antibodies that had a bivalent intraspike binding, which the authors of this paper basically concluded that this is why they were able to inhibit multiple variants, including Omicron. And we've, over time, these never made to the clinic, but over time we keep looking at different variants, and these antibodies have really lasted to even be BA4 and BA5 were the last ones that we looked at, and they still inhibit at really high, at really low IC50s. So that's basically the COVID story, and there's a lot more data, and that COVID story is published this year if you want to read a little bit more about that. The other disease that we wanted to look at was influenza. And as all of you know, there's two dominant proteins of influenza on the surface. There's imagglutinin and neuraminidase. So basically, we wanted to see the challenge with influenza, obviously, is that, again, like COVID, it's very variable. You have to find these antibodies that it's been shown that if they bind to the stem, they can cross-neutralize. So our goal was basically to find antibodies that cross-neutralize multiple variants of influenza. So how did we do this? we had 2 studies going on because we didn't quite know whether we needed people that have been recently immunized, or if we used just previously immunized individuals, we will still find these antibodies. But the antigen that we used to sort the B cells was kind of our secret here, where we basically thought if we want to have cross-neutralizing antibodies, we should look at antigens that we don't normally see. So if we're using N1 and N2, we'll probably find a ton of antibodies. But if we go in and say, okay, I want antibodies to I want B cells that recognize H7 and H5, those antibodies are most probably going to be cross-neutralizing. And with NA, we went with a little bit less, more conservative approach where there's not a lot of studies out there that show neutralizing antibodies to NA, so we went with just N1 and N2, knowing that if it works, we were going to go with, again, the same strategy that we're using for HA. So this was, again, extremely successful. This was a lot smaller study. I think we learned a lot from COVID that we don't need to look at 300 people. So with this study, we looked at 10 to 15 individuals. And we were able to isolate about 15,000 antibodies. We made close to 500 of those antibodies. And majority of the antibodies bound to H1 and H3, even if we did isolate it with a different, basically different antigen. And then we had 38 of those antibodies that were cross-reactive between the two. And we had 13 antibodies that actually bound to all HA that we tested so far. So that, to us, that was pretty successful because those antibodies are really rare to find. And with the NA, again, that was a way smaller study, but we did find antibodies that bind to both N1 and N2. As you can see on the next slide here with the HA, so we basically went in and sourced a bunch of HA proteins from HA1 to HA10. And as you can see, these antibodies bound, our lead candidates bound with really decent affinities. And then, this was done last year, and as we were doing this study, there was a lot of partners that really wanted to know a little bit about what's going on with H5, because that's what was going on at the time with H5N1 in birds. So we went in again and just sourced a lot of antigens to H5 from different, over a long period of time. I think we went, I can't see there, but up to 2007. And as you can see again, our lead candidates bound to all different kind of H5 that we did look at. So this is the summary of a lot of data when it comes to neutralization. And here we're comparing it to MEDI8852, which a lot of you know has been published and it's been shown to cross-neutralize. And what I didn't talk about is that when we did epitope mapping of these antibodies, they actually, majority of them bound to the stem. So it was really interesting that we were able to find these diverse set of antibodies, and using our bioinformatics and our technology, we were able to pick up these really cross-neutralizing antibodies. And again, on the right there, you can see that we went and did pseudovirus for a few other H5s, and they neutralized at really low IC50s. So again, really successful with COVID, again, with flu. And with the NA, we only found 2 antibodies, and these antibodies actually belong to the same lineage. But again, we didn't really, with NA, we didn't go really hard on this, and we were able, in the process of producing a couple of N5 and N7 to be able to use it in this assay and do a lot more sort from a lot more individuals. So here at the top, you can see the ELA assay, and this is using live virus. And basically what's happening here is we can block the ability of NA to cleave sialic acid. And we're using 1G01, which was published a few years ago as a control, and our antibodies perform really similar. And then we also did neutralization here at the bottom, and if you've followed flu, it's really hard to find this neuraminidase neutralizing antibodies. I think VIA has one that's gone to the clinic, but there's very few of them, and we were able to find in this very, very small study antibodies that neutralize NA. So as I said at the beginning, one of the big advantages of pair-seq is that we can basically pair millions of cells at a really short time. And once, the disadvantage of that is you have a ton of sequences. And what do you do with that? So basically, we will put them in these lineages. And for flu, for example, we did find an antibody right there highlighted in black that we made in that first synthesis. And then usually we will say, okay, which antibodies are the most neutralizing? Can we go back and do they have lineage mates? And this was really interesting because this antibody had about 32 lineage mates. And we were able to go back in and pick a few other antibodies and say, can we find an antibody that's better than that first one that we isolated? And this is really important because basically we are allowing the immune system to affinity mature these antibodies. Otherwise, we'll have to go after that one antibody and do affinity maturation. So that's basically what we've done so far with pair-seq. We believe it's really robust. And I didn't talk about the humanized mouse project that's going on this year. And at this point, we're going after targets that are a lot more difficult. Obviously, we've worked with infectious disease, but that's really not our specialty. We want to go into autoimmune, go into cancer. And we've been able to pair over 180,000 antibodies over the last few years and validated about 2,000 of those antibodies. So just to summarize, basically our true AB discovery system can generate hundreds of thousands of naturally occurring, fully human antibodies with that really high speed, high quality data set with really deep breadth, and we can go back and always find newer antibodies from the data set. This really also helps with our bioinformatics and basically being able to train the data set with a ton of data that we know either bind or don't bind. Anyway, so this is kind of the data set. I don't have a thank you slide, but I just want to thank everybody that's worked on this project, mostly from Adaptive and GenScript for making all our proteins at really quick speed. And also some of our collaborators, like the COVID project. And I'll take any questions. Yes?
I'm a little bit curious about how did you achieve, maybe that's part. I'm very excited and curious about how did you achieve actual pairing of the natural high-value antigen from thousands of the cells in that well.
Yeah, so we can put basically, we can put as many, we've done this study where how many cells can we put in a well? Because that's your limitation, right? What I've found is that if I go over 30,000 cells a well, I will break the system. And I think it's mostly because of reagent. It's a reagent problem where I'm probably running out of reagent at that moment. So my pairing will start going down. So that's how we're able to do that. I normally do about 5 to 10,000 cells. So if you can imagine, that's 5 to 10,000 times 96 wells. So that's how and we're not limited to one plate. So we can do up to 10 plates per sequencing run, basically. So that's how we're able to get those millions of cells at one time. Any other questions? Okay. All right. Thank you.
I hope I got that right. Senior scientist at GenScript.
Hi everyone, so I'm Heng Tai, senior scientist from GenScript, and it's great to see you here today. So today we are going to talk about the antibody fragments and from their therapeutic applications to a robust production as well. So the talk will be actually divided into 4 sections. So we'll actually first come through about the overview of the antibody fragments and their therapeutic applications. Then we'll talk about the challenges in antibody fragments production. Then we'll also talk about the therapeutic platform, which is a very robust platform for the faster antibody fragments production, and of course the antibody. So, and then after that, we'll make concluding remarks for the whole talk. So first, we give a very quick introductions. So GenScript Biotech Corporation is actually the world-leading technology and service provider of life science R&D and as well as the manufacturing as well. So built upon its solid gene synthesis technology, so GenScript Biotech is actually divided into 4 major platforms. So as you can see over here, so including the life science service and the product platform, so, and the biologics contract development and manufacturing organizing CDMO platform, the industrial synthetic biologic products platform, and as well as the cell therapy platform. So, GenScript was actually founded in New Jersey, United States in 2002. So, and now our business operations actually spanning over 100 countries and regions worldwide as well. So because as I'm from the GenScript life science service platform, so focusing in the mammalian protein and the antibody protein production. So today I'm going to share more about the antibody fragments production and its challenges we encounter and how we deal with that. So first, we will have some overview of the antibody fragments and their therapeutic applications. So what are actually antibody fragments? So the antibody fragments are the fragments of the binding end of an antibody. So although it can be very small, but it's a very strong and a very powerful tool to fight against the disease like antibody. So here's actually some very common members of the antibody fragments. So such as the Fab, the VAH, so the SCFV, and as well as like the mini, di, tri, and tetra body. So we know that the antibody fragments like Fab and the SCFV, etc., is actually much smaller than the full-size IgG. So, and thus they show a very greater cell penetrations, and let's say in the solid tumor as well. So for example, Lucentis, so which is a VEGF-A specific Fab fragment, was actually more effective than their corresponding full-length monoclonal antibody Avastin. So in treating patients with the wet form of the macular degeneration. So we know that antibody fragments, let's say like Fab, has a shorter half-life due to the lack of the Fc receptor mediating recycling. However, the half-life can be easily extended by conjugation to, like, let's say, the PEG or fusion with the proteins like the albumin-binding protein. So, and the related drugs that FDA actually has approved is like, let's say, the certolizumab, PEGO, so pegylated and humanized anti-TNF-alpha Fab. So for the treatment of the Crohn's disease and as well as the rheumatoid arthritis. So also here's the quick summary of the FDA-approved antibody fragments, so with various formats and their targets. So a few more recent FDA-approved nanobodies such as the capilacizumab. So it's also involved in the treatment of the acquired thrombotic thrombocytopenic purpura, or we call the TTP. So what is actually TTP? So it's actually a very rare blood disorder. So that results in the blood clots in the small blood vessels and around your body. And these clots can actually slow the flow of the oxygen-rich blood to your heart, brain, and other organs as well. So the disease affects the platelets, blood cells that stick together to help your blood clot when you actually cut yourself. So they also seal the cut and preventing the bleeding. So in the TTP, so the platelets actually they clump together and they form a small clots when they shouldn't. So this leaves the fewer platelets in your body, so which is why you may bleed more than usual. So here's actually the mechanism and how it actually causes the disease. So let's get back to the drug, the caplacizumab. So the caplacizumab, how it actually works to fight the disease is actually, it actually blocks the interaction between the VWF, as you can see over here. So the VWF is not, okay. So the VWF, which is the green, the string. So, and as well as the platelet glycoprotein. So these will actually block the adhesion and the clumping of these two compounds. And this can actually prevent the microvascular thrombosis and as well as the organ damage as well. So here is actually the power of the caplacizumab. So, and then another interesting FDA-approved SCFV molecule, which actually target the eye disease, or we call the macular degeneration. So it's actually the drug brolucizumab. So as you can see, so as we know, the wet macular degeneration is an eye disease that can actually blur the center of your vision, which is shown in the figure. And it's a very serious type of the late macular degeneration. So it happens when the protein called the VEGF make the abnormal blood vessels grow in the wrong place at the back of your eye. So due to the lower molecular mass of this drug, so compared to their larger counterparts like the ranibizumab, so along with their fantastic solubility, allows the production of the solution with a very high concentration, which can actually up to 122 milligram per ml. So this level of concentrations, they actually can allow the administration of at least like 6 milligrams of the drug in a standard intravitreal injection. So, yep. So again, the key question is like, why antibody fragments? So because of their lower non-specific binding from the Fc interactions, and more importantly, the antibody fragments can be used to eliminate the Fc-associated effector functions in the antigen-antibody binding studies. So also, it can also be used to achieve a lower immunogenicity than in the intact antibody for the experiments in vivo. Also, more efficient penetrations of your tissue sections, resulting in improved staining in the IHC. Okay, so now we look at the challenges in the antibody fragments production. Either in lab or in the industry. So the challenges, so there are challenges when producing the antibody fragments. So in this case, I would like to focus more in the SCFV. So because we know that the CDR, the CDR regions, they are actually responsible for the antigen binding. They can also contribute to the aggregation due to the frequent occurrence of the hydrophobic and the electrostatic residues as well. So also, the VH field interface of the SCFV is usually quite dynamic, and they can also experience the transient opening, which could expose the hydrophobic patches to promote the aggregation. So SCFV is actually very challenging when we produce. So therefore, how to actually stabilize the SCFV through the modifying the VH and VL interface is actually not the main topic that we are going to discuss today. But today, what we are going to talk about is like how we actually find that the aggregation can be a challenging issue in the productions of these SCFV containing multi-specific antibodies. So we will use this whole set of the SCFV containing antibodies as an example. So of course, so bear in mind that the expression system that we are going to discuss today is the mammalian expression system, or particularly the CHO. So the production of the series of the scFv containing multi-specific antibody, so you can see that resulting in various EU. So with the blue indicates a very high or moderate expression level. So while the red indicates the low expression level of the scFv containing antibody. So interestingly, so we also observed that those high expression level panel, they usually have no issue of aggregation during the production, while those low expression panel, they actually have a very serious issue of the aggregation, as you can see, which is over here. So most of the products that we get is actually the aggregate products, while the blue one, so with the high expression level, they usually is good in quality and no aggregation. So in this case, we thought that this aggregation and the low expression of these scFv containing antibodies is actually might be caused by the scFv, or we call the problematic scFv, scFv1. So then we actually try to swap it into another scFv, so which is we call the scFv2. So as you can see that from these results over here, so the U actually improved significantly after swapping the scFv. So this actually comes to a conclusion that even they are both scFv, however, due to their characteristic and probably due to the CDR region or the framework regions of the scFv, so these might actually result in a very huge difference of the U and as well as the aggregation, which we are going to talk about now. So you can see that after swapping the scFv, so you can see that the aggregation issue is actually resolved perfectly after that. So you can see that the scFv2 results in almost no aggregation, almost 100% of the target antibodies. So here's another example. So you can see that the aggregation issues actually resolved perfectly. So after swapping the scFv. So this actually brings up one of the major challenges in the antibody fragments production, which is the aggregation. And this might be caused by the CDR regions of the scFv and as well as the framework region. So it brings to the second challenge where the design and also the sequence of the framework regions might affect the expression and the aggregation level. So in this case, we show that a wild-type antibody with extremely low expression level, so you can see that which is about 1 milligram per liter. So they also have a very serious aggregation issue here as well. So through the sequence analysis, so we actually found that the antibody is missing a tyrosine 36. So in the light chain, where the tyrosine is actually very likely to be involved in the folding and stabilizing of the antibody. So after insertion of the missing tyrosine 36 back to the antibody, so from here you can see that the aggregation is actually significantly improves and then also resolved perfectly. As you can see that the expression level also increased a bit. So now we are going to talk about one of our very robust platform for the antibody production and of course the antibody fragments. So we know that recombinant antibody expression and also the antibody fragments in the mammalian cells is actually very crucial when the post-translational modification and appropriate folding are desired for the downstream application. So commonly, the top challenge of the antibody expression projects are the insufficient U of your antibody, so and also a very long time for the completion, like months. So and as well as most important thing is the budget. So to lift up all these bottlenecks, so GenScript actually has launched its proprietary turbo platform, so which actually offer a higher U performance within a shorter time frame, so which is designed from small, medium to very large scale production. So and recently, our turbo platform actually has been successfully upgraded to 2.0, so which actually unlocked the shorter timeline and higher yield performance of the antibody. So of course, you can approach to our booth for more information. So like I say in the previous slide, so the TurboCHO 2.0 aims for even faster delivery, as fast as 8 business days. So starting from gene synthesis to the delivery of your antibodies. So yeah, I think I will skip the TurboCHO 1.0. So we'll straight away jump to the 2.0. Yep. So from this, you can see that we show that with the TurboCHO 2.0, so the yield actually obtained was actually much higher with different formats of the antibody fragments. So here's some of the antibody formats we are actually trying. So we can, from the right panel, you can see that, which is we actually compare the yield between the 1.0 and 2.0. So you can see that from a 2.0, the yield actually increased significantly. So here are also like some other very different formats that we try, including the bispecific antibody, and as well as like the BITE format and the knock into whole format of the bispecific antibody, which all shows that the expression level increase, like even up to tenfold. So that means that from this service that we actually provide a shorter time frame and also a higher yield for your protein. So yeah. So for the conclusions, and you can see that we actually like discuss the antibody fragments and some of their therapeutic applications, and also like the challenges in the antibody fragments production, which is the aggregation issue, and sometimes it can be caused by the framework region and your CDR region as well. And then also we also talk about like the TurboCHO platform, which is a very robust platform for the faster antibody fragments production. So which actually has a very short time frame. It's like about 8 business days, and then which also have a very high expression level compared to the 1.0. So this is basically all my talk for today. So yeah. So thank you very much. Okay, yep.
Quick question. So the difference between the TurboCHO 1.0 and 2.0, is that the aggregation modification, or is that something, have I misunderstood?
Oh, yeah. So you are asking like regarding this slide, right?
Yeah, what was the improvement,
so,
I guess?
for the, that's a very good question. So for the 1.0 and 2.0, in this case, we actually referring to the CHO. So, and then for this CHO, we we actually in this 2.0, we actually upgrading the cell line, the vector and all this thing, and then to improve the yield. So in this case, like what you actually are asking, like whether they solve the aggregation. So I would say like to a certain level, but the aggregation issue of the antibody is still depends on the characteristic of the antibody. Yeah.
So.
Yeah.
So, I have a validated antibody sequence. So what do you need from my, full antibody to make the fragments?
So you mean like you give us the antibody?
Yeah.
And then
if I give a sequence, you're able to make it the fragments for me?
Yeah, we can actually like make the fragments. Like if usually you are regarding like if you can just straight away send us the sequence of your antibody or the antibody fragments. So then we can produce from that. But sometimes we can actually like start from the monoclonal antibody. Then we can actually like cut it and then to make the fragments, like for example the fat itself. Yeah.
Thanks.
Hi, I liked your story about the tyrosine in the framework. Now, since you're seeing a lot of single-chain FVs, I was curious if you're employing any type of machine learning bioinformatics to, from the beginning, look at the sequences of single-chain FVs to try to predict and learn how they will behave, and maybe suggest some changes or optimizations.
I mean like whether we actually perform the bioinformatics to study, whether how it affects...
from the sequence.
Or from a
for
sequence.
sequence motifs, or trying to see any correlations from the assays that you're doing, such as analytical SEC, probably
that's
stability.
a very good
then
question.
kind of learn from.
Yeah, so we actually do have a certain AI projects at this moment. So also like studying how the sequence, like we're studying like tons of a sequence and try to find the correlations between like the sequence and as well as the aggregation level, the expression level, all this. And it's actually, we are still doing that. Yeah, so, but that's a very good question. Yep.
I'm curious about, like, since you mentioned, once you see some aggregated SFV, it will probably be a result of the framework or the CGR regions. So what protein engineer techniques you guys will apply to tackle those problematic framework or CGR regions? Do you try to swap onto different framework? Do you change the orientation, optimize with the linker? So other than cell lines, what other techniques can be necessary and helpful for this overall aggregation issue?
Yeah. Oh, that's a very good question. So because like once we actually get a sequence, so we actually like have our software. So to actually try to actually fish that is there any place which is like suspicious, whether like this mutation, because like sometimes the clients, they tends to do mutation within the framework region as well. So this might actually disrupt the folding or the interactions between the let's say the alpha helix or the beta sheet, all this. So we actually do have the software so to predict that. So after that, we actually like can suggest whether they need to do the mutations to switch it back or not. Yeah. Yeah, but that's a very good question. That's a very good question. Yeah.